Chapter 7, Part 3, looking at proteins and some additional forms of allowing things to cross the membrane. We're going to start by looking at facilitated diffusion, which is almost identical to diffusion. The solutes are moving down the concentration gradient. We can see that there's a higher concentration of the solutes on the top part of this picture and fewer on the bottom. So we're going from high to low concentration gradient. This is going to stop at equilibrium, and it's going to be occurring passively. The one difference that we really have between facilitated diffusion and diffusion is a protein channel is required. Hence the name facilitator, to help. So to help these solutes cross the membrane. Now, facilitated diffusion can occur with different types of proteins. One would be a channel protein mentioned in the second part of Chapter 7. We can see the channel. And the second one would be a carrier protein, which is a protein that helps things cross and in the process changes shape. And we can see here that it clearly has changed shape to allow this to cross. Quite modest with the change of shape. It's a bit more intensive than that, but this sort of illustrates the idea. Some examples of the channel proteins would be those aquaporins for water to move through, and also proteins specific to various molecules or ions. Some channel proteins have gated channels or ion channels that open and close in response to a stimulus. All right, moving on. And now we have active transport. As the name suggests, it is not a passive process, and therefore requires some energy, or ATP. Active transport requires energy because, unlike the passive transport, active transport goes against the concentration gradient. Because of this, active transport will not reach equilibrium. And the example we'll use is the sodium-potassium pump, which is used by our neurons to relay impulses. Starting in image 1 right here, and working our way clockwise, we can see a protein that is collecting some sodium ions, which are the NAs, and then along comes our boy ATP right here. ATP will phosphorylate this protein, or donate a phosphate, causing the protein to change shape, and it will pump the sodium ions out of the cell. In this case, we have the extracellular fluid inside of the cell, so this would be outside the cell. Then along comes potassium, and potassium will go across the membrane in the opposite direction of the sodium. Notice in step 4 right here that the movement of potassium does not require ATP, but it does release the phosphate donated by the ATP molecule in step 2. So this phosphate is released. The phosphate will later be picked up by an ADP molecule, and that would revert back to the ATP state. So to recap a little bit, diffusion is a passive process. It moves solutes down the concentration gradient. These solutes would be non-polar. They can go straight through the bilayer. Other forms of passive transport would be facilitated diffusion, which typically will occur for those polar substances which cannot simply cross through. They'll still move down the concentration gradient, have a couple different types of proteins, whether it's the channel or carrier protein, from high to low concentration. And then we have active transport, which moves solutes against the concentration gradient, and because it's kind of an uphill process, we have to phosphorylate a protein to dump off some energy for the solutes to cross. This image shows us cotransport. Cotransport is a special form of transport that involves a little bit of coupling and is a form of active transport. Our example illustrates the necessity of a proton gradient the protons being the hydrogen protons in this case. And these hydrogen protons will help move molecules into the plant cell, sucrose being the item in this particular case. So the only way that sucrose can get inside of this particular cell is through this protein right here. And the only way this protein will move, or move the sucrose into the cell, is if there's some protons available to bind into this site right here. So the proton pump will use ATP to move the hydrogen protons to the exterior of the cell to create concentration of these, which will then be used to drive the sucrose back into the cell. So it's a coupled reaction. This process right here cannot occur without this process occurring as well.
Next we're going to be talking about endocytosis and exocytosis. We talked a little bit about endocytosis previously. Endo meaning into, exo meaning out of. So endocytosis is taking things into the cell, things that would be too large to go straight through the plasma membrane or through those protein channels. These would be the macromolecules, proteins, carbohydrates, glucose. They're too big. So what happens is these food products would approach the cell membrane, and the cell membrane would kind of pinch in a little bit or indent, and then the food molecules would go into that indentation where the membrane would kind of pinch off a vesicle containing the food, which would then now be inside the cell. And we spoke previously about how these vesicles would then be attacked by the lysosomes, and the food would be broken down. Three examples that we're going to talk about for endocytosis are phagocytosis, pinocytosis, and got a typo here. This should be receptor-mediated endocytosis. Exocytosis, on the other hand, is the exact opposite. This is going into a cell, so exocytosis would be removing from a cell. Still uses those transport vesicles. It's just to get things out of the cell. The things that would be leaving the cell, well, it varies. One example would be waste products. Other things would be included in there as well. Just really depends on the size of the item, if it can simply go through the protein or not. So this image is showing us phagocytosis, which pretty much is exactly what I was describing on the previous slide, where the food particle needs to get into the cell, but it's quite large. So we have this little indentation, the food is going into it, and then we have this little vesicle being broken off, and now it's inside the cytoplasm of the cell. We can almost look at this as wolfing down a burger. You eat it really quickly, you take huge bites. This is a little bit different than pinocytosis. Although pinocytosis is similar to phagocytosis, but the particles are not wolfed down as much. We can kind of see this guy's taking a huge bite, actually almost sticking his mouth out a bit. And this guy's kind of accepting stuff as it goes in, kind of like it's slurping in the cell with some of the extracellular fluid and some of the solutes within that cell solution outside here. Once again, we have the vesicle inside of the cytoplasm. And the last thing we're going to be talking about are the receptor-mediated forms of endocytosis. These involve the use of ligands. These are the Y-shaped things right here. The receptors and the ligands are the triangles that fit into them. So receptor-mediated, they're controlled by these receptors. Now these receptors are specific to the ligand, and with the appropriate ligand in place, we'll have the indentation and the accepting of the cellular fluid to form one of those vesicles once again inside of the cell. We can see it kind of going on right here. It's a little grainy, but nonetheless you can kind of see the pinching off of the vesicle that's forming. 25 nanometers, quite small.
So this will conclude Chapter 7. Chapter 7's a bit shorter. Take it as a break. We'll be moving into Unit 4 shortly.